Good morning, students. Today in this session we are going to start first part of cell, the unit of life of 11th standard. In this we will discuss about cell theory, an overview of cell, prokaryotic and eukaryotic cell envelope and its modification in prokaryotes, gram-positive and gram-negative bacteria, pili and fimbriae, ribosomes and inclusion body in prokaryotes. First of all, we will know about cell. What is cell? Cell is the smallest structural and functional unit of life. A single cell is capable of independent existence and also performing the essential functions of life. Anton van Leeuwenhoek first saw and described a live cell of bacteria. The term cell was introduced by an English scientist, Robert Hooke. He examined thin slices of cork under a crude microscope assembled by him. He observed tiny compartments in the slices and he called them as cellulae, now termed as cell. The branch of science that deals with the study of cell is called cytology. In the given figure, you may observe the microscopic structure of a thin slice of cork tissue showing tiny cells. It is the primitive and crude microscope assembled and used by Robert Hooke, and its magnification power was 14 to 42 times. Now it is about cell theory. The cell theory is one of the most fundamental generalization of biology. It was proposed by Matthias Skeladon and Theodor Sachwan in Plants and Animals respectively. Cell theory states that all living organisms are composed of minute units called cell and cell products. Cells are the smallest entities that can be called living. All the cell arises from pre-existing cells by cell division. Rudolf Virchow first explained that cell divided and new cells are formed from the pre-existing cell. That is Omnis cellulae e cellula. Now let us see about an overview of cell. A cell has three major components. What are they? They are cell membrane, also called as plasma membrane, cytoplasm and nucleus. The cytoplasm and nucleus further have several components. All the components of a cell taken together keep it living. Plant cells, most fungi and certain protists have a thick rigid non-living additional covering outside the cell membrane called as cell wall. It helps in protecting the cell. Nucleus is the largest and most prominent spherical organelle in eukaryotic cells, which is bounded by a thin double membrane called as nuclear envelope or nuclear membrane. Nucleus contains chromosome and chromosomes contain DNA, and this DNA is the genetic material. In the cytoplasm of eukaryotic cells, different membrane-bound organelles like mitochondria, endoplasmic reticulum, Golgi complex, lysosomes, microbodies and vacuoles are also present. Ribosomes are non-membrane-bound organelle present in both prokaryotic and eukaryotic cell. Ribosomes are also present in chloroplast, mitochondria and rough endoplasmic reticulum. In animal cell, centriole is also present, which is a non-membrane bound and helps in cell division. Cell differ in size, shape and function. Altogether, when we consider smallest cell is mycoplasma and largest cell is egg of ostrich. In human being, smallest cell is red blood cells and largest cell is nerve cell. Here you may observe overall smallest and largest cell as well as the smallest and largest cell in human body. Now let us discuss about differences between prokaryotic and eukaryotic cell. There are so many differences. When we consider the size, then prokaryotic cell is a small, range between 1 to 10 micrometer, whereas eukaryotic cell is large as compared to prokaryotic cell, and it is in between 5 to 100 micrometer. In case of prokaryotic cell, nuclear region is not well defined and not surrounded by a nuclear membrane and are called as nucleoid. Whereas in eukaryotic cell, there is well defined nuclear region and are well defined as well as surrounded by nuclear membrane. Chromosome in case of prokaryotic cell is single and circular. This is very important. What? In case of prokaryotic cell, chromosome is single and circular. But in eukaryotic cell, chromosome is more than one and are linear. No membrane bound organelle are present in prokaryotic cell, whereas membrane bound organelle like mitochondria, Golgi body, endoplasmic reticulum, etc. are present in eukaryotic cell. In prokaryotes, the cell division is by simple fission, that is by amitosis, whereas in eukaryotic cell is dividing by mitosis and meiosis. We will study these two terms in the another chapter. Prokaryotic cell lack two nucleus, but in eukaryotic cell, two nucleus is present. In prokaryotes, ribosomes are randomly scattered within the cytoplasm, but in eukaryotic cells, ribosomes are bigger and are attached to the endoplasmic reticulum or freely seen in the cytoplasm. Examples of prokaryotes are bacteria, blue-green algae, mycoplasma, that is PPLO, pleuropneumonia like organism, and examples of eukaryotes are protists, plants, animals and fungi. Here in the given figure, you may observe the prokaryotic and eukaryotic cell. Here it is also about prokaryotes and eukaryotes. Most of the prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. So in this slide, we will discuss about the cell envelope and its modification in prokaryotes. In case of prokaryotes, the cell envelope consists of tightly bound three-layered structure. Outer layer is glycocalyx, middle is cell wall, and inner is plasma membrane. Each layer performs distinct functions and they act together as a single protective unit. Glycocalyx differ in composition and thickness among different bacteria. In some cases, it may be a loose sheath called as slime layer, while in another it may be thick and tough called as capsule. The cell wall determines the shape of the cell and it provides a rigidity, strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is semi-permeable in nature and interacts with the outside world. This membrane is similar structurally to that of eukaryotes. A special membranous structure is the mesosome, which is formed by the extension of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, then lamella, which helps in cell wall formation, DNA replication and distribution to daughter cells. They also helps in respiration, secretion and to increase the surface area of the plasma membrane and also enzymatic content. In some prokaryotes, for example in cyanobacteria and other membrane extension, are also present in the cytoplasm, and they are called as chromatophore, that contains pigments. The bacterial cell may be motile or non-motile. If motile, then they have flagella. Flagella consist of three parts: filament, hook and basal body. And flagella helps in locomotion. Besides flagella, pili and fimbri are also present on the surface of the bacteria. So they are also called as surface structures, but they do not play in the movement. Only the flagella is playing role in locomotion. Now we will see what is the difference between gram positive and gram negative bacteria. Some bacteria take up gram stain and others not. So those bacteria which are able to take up gram stain is called gram positive bacteria, and those bacteria which do not take up the gram stain is called gram negative bacteria. In this figure you may observe that in gram positive bacteria the purple crystal violet stain is trapped by the layer of peptidoglycan, which forms the outer layer of the cell. So what? It is trapped by the peptidoglycan, and this is the outer layer of the cell. This is the microscopic view of gram positive and gram negative bacteria. Now let us see about the structural difference between pili and fimbri. The pili are enclosed tubular structures made up of a special protein, whereas fimbri are small bristle-like fibers sprouting out of the cell. In some bacteria, these structures helps them to attach with rocks in streams and also to the host tissues. In the given figure, you may observe the cell surface appendages of a bacterial cell. You can observe here fimbri, pili as well as flagellum. Here there are the different forms of bacteria: cocci, bacillus, spirula, vibrio, etc. Now it is about ribosomes and inclusion body in prokaryotes. In prokaryotes, ribosomes are associated with plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size. Ribosomes in prokaryotes are made up of two subunits, that is 50S and 30S units, which is when present together they are 70S. Ribosomes are the site for protein synthesis. Many ribosomes may attach to a single messenger RNA, that is mRNA, and it forms a chain called as polyribosome or polysomes. In prokaryotic cell, reserve material are also stored in the cytoplasm in the form of inclusion body, which are not bounded by any membrane. So that is why they are called as inclusion body, and lie free in the cytoplasm. Examples of inclusion bodies are phosphate granules, cyanophycean granules, glycogen granules, gas vacuole, which is present in blue-green alga, and purple and green photosynthetic bacteria. So this is all about today's session. Thank you. For any kind of doubts, please write your question in comment section.